All right, folks. Let's go ahead and get going. Thanks so much for coming out tonight. I know also a lot of folks are in town for PEGS. So for everyone who's not from Boston, welcome to Boston. Really excited to see so many folks. It's really like such a great energy in the room as well. Just want to start by thanking all of our sponsors. You can see by looking around in the room, there's a lot of folks, a lot of mouths to feed. These are the folks that provide the pizza and beer. I would also just request, we know we have a lot of folks from Big Pharma in the audience, so we rely on the support of the community to make sure we can keep this going. So if you're at a Big Pharma, please reach out to the people that control the money and ask them, hey, can we donate? It's not super expensive and you can get your logo up here. So I'm going to be reaching out also to the people in Big Pharma who've emailed me, and you know who you are, so expect an email. It's really the generosity of the community that keeps this thing going. So yeah. So I'm very excited to have our speaker also from out of town. So Ariel Tennenhouse. So he did his undergraduate studies in Berkeley. He's from San Francisco originally, and just defended his thesis with Sorel Fleischman at the Weizmann. So freshly minted PhD. Ariel, take it away.
Okay, let's just figure it out. All right, hopefully that's working. You wanna?
We're just thinking about the lines.
Oh, okay. That looks promising.
Yeah, we'd like.
Okay, yeah. So thanks, Chris, for the really nice introduction. We're sitting in the Weizmann Institute, a bit outside Tel Aviv, and we don't really have too much of a protein design community around us. So I've always been really, so I've always been really envious of this club, and especially since Ariel gave a talk here about a year ago when he was at PEGS. Had really amazing things to say about his time here. So I'm really happy to be here to kind of give an update about what we've done in the last year, and maybe it's also interesting to hear my perspective on the work from a bit closer than Sarel sits. Can I go to this? Someone tell me when I'm on this. Okay, so yeah, antibody therapy. Let's just leave it. It's okay, right? So yeah, antibody therapeutics are now, of course, used to treat a wide range of disorders, ranging from things like cancers to neurological diseases to autoimmune diseases. And there's more than 130 antibodies, of course, that have been approved by the FDA for clinical use up until now. And that number is, really skyrocketing. I think that 130 is already probably quite a bit out of date. And that's really a tremendous accomplishment built on the backs of works of tons of really great scientists and antibody engineers over the last few decades. That being said, it remains difficult to develop antibodies into therapeutics for many reasons, but one of the reasons for that is that in order to be useful therapeutics, antibodies need to excel in many characteristics, including things like humanness, or you could think of that as sort of not causing adverse reactions because of immunogenicity reasons in patients, and also including things like affinity and stability. And one of the really tricky things about this is that these characteristics are not independent of each other, and some of them are actually even really oftentimes inversely correlated with each other, with mutations that, for example, might improve antibody affinity, oftentimes degrading antibody stability along the way. And these kind of trade-offs are just not acceptable when we're talking about therapeutics. And yet, antibody optimization pipelines often still utilize iterative experimental approaches. And because of these inverse characterizations, these can lead to trade-offs between these important characteristics, which gets you into these kind of long optimization cycles. And maybe kind of more near and dear to my heart, using these sorts of experimental approaches really admits gaps in our understanding between the links of antibody sequence structure function relationships. And I show stability here as a thermostat. A lot of people would think like the first thing that comes to their mind when talking about stability is thermal stability. But of course, I think stability really encompasses much more than thermal stability, also including things like expression yield and purity, resistance to aggregation or self-association, hydrophobicity, polyspecificity, and polyreactivity. And in the last few decades, all of these kind of characteristics and also other things have been sort of characterized under this umbrella term called developability. And I'll just use this sort of umbrella icon to refer to developability throughout the talk. And despite really an enormous amount of effort going into developing methods to characterize developability and predict developability, predicting or optimizing antibody developability, I think actually remains quite difficult. And this was shown, I think, actually quite convincingly in two recent papers, one preprint from Jeff Gray's lab, and another actually published paper now from Ginkgo, where they basically showed that at least with publicly available things, it's very difficult still to predict antibody developability. And so because of all these reasons, expert teams in industry spend on average, I think now 2 years optimizing leads before preclinical studies. Of course, this is just kind of one piece in the puzzle, but this is a contributor to the long development times for antibody therapeutics and the eventual, high cost for patients. And so I think one goal for computational design in this space should be, to enable faster optimization and to better manage these multi-parameter trade-offs. I think that's a very kind of realistic goal for computational design. And if it can be done using, principled computational approaches, that, I think would be really great. And I think that's a great goal for the field. Taking a step back a bit from antibody optimization, there's really, I think, two main pipelines for antibody discovery these days. One is to immunize animals with the target antigen, and the second is to display synthetic libraries of human antibodies on the surface of phage or yeast, which can then be screened for binding to the target antigen. And these approaches really have a lot of potential benefits over immunization in terms of the control over the process and also the speed at which they can be done. And for these reasons, phage display was given, half the Nobel Prize in 2018. And yet, despite these really potential significant advantages of synthetic libraries, 90% of the approved therapeutics in the last decade, according to a report that came out from the European pharma companies, were discovered using mammals or animals. And so you can, start asking the question of why is that, given the, potential huge advantages that come from using synthetic libraries. And at least I think one explanation for that is that most of these synthetic libraries are formed in one way or another by randomly combining either antibody frameworks or mutations on top of antibody frameworks. And these random or semi-random processes that are used to construct these repertoires do not address critical foldability and stability rules. And therefore, they may produce antibodies which are very human and have high affinity, but don't have such good stability or developability. And therefore, you've sort of solved part of the problem and you've done it faster, but you have to do so much work on the back end optimizing these antibodies that you haven't really per se taken a step forward. And in the end, maybe it's actually easier to use antibodies that come out of animals. So during my PhD, we set out to develop a strategy to design synthetic repertoires of antibodies that are both developable and structurally diverse. And so there's two parts to that sentence that I just said. Yeah, so first, we're a Rosetta lab, right? So we have to have an acronym for everything. So the acronym for this project is CADABRA, Combinatorial Assembly and Design of Antibody Repertoires. We wanted to make it like abracadabra, but we couldn't figure out the acronym for it. So this is what we ended up with. And so there's two kind of points to that sentence that I made, right? The first is developable. And I think that sort of speaks for itself, but there's a question of how can we go about doing that? And so we thought that, given that predicting or optimizing developability is sort of difficult, we could start by at least trying to optimize the native state energy of all the antibodies that are in our library, with the idea that if we can make these libraries sort of really structurally stable, then at least you don't have, loops flopping around that can lead to all kinds of non-specific binding and aggregation. And in order to do that, we wanted to be able to do this with absolutely no randomization. So every antibody that's in these repertoires is completely designed. And, modulo some PCR errors here or there, we really know everything that goes into these repertoires. The reason that we thought this might be a good idea, it might sound crazy, is that there's been a lot of work, both from our lab and from other labs, showing that structure-based methods can successfully optimize antibodies and other biologics for a variety of different kinds of properties, ranging from stability and expression to humanness to affinity. But this has been done mostly on a case-by-case basis. And so kind of one of the main goals of my PhD was to think about how can we extend this, from the case-by-case basis to the repertoire level. The second part of this kind of goal statement that I put out is that we wanted to have high structural diversity. And the reason we thought this is really important is there's, of course, been a lot of effort put into how to make better synthetic antibody repertoires in the last years. But most of these efforts have worked by really reducing the amount of frameworks that goes into these libraries, building them based on frameworks that are sort of known to go very well through clinical trials or known to be highly developable. And we think that this is a super successful approach and has led to repertoires that produce much more developable antibodies, but this may also kind of limit the eventual structural diversity that you get out and therefore the epitopes that you can target. So we thought that a complementary approach that is able to use a lot more frameworks because of all the design we're doing on the front end could also be very valuable. And just to kind of say that I think there's still really a place for antibodies in the world of de novo design. De novo design has really made tremendous progress in the last years, but I think a lot of that has been in targeting hydrophobic epitopes. This is just to show, for example, this is, I'm showing here a binder that Ariel designed in 2011 against influenza hemagglutinin. And there are two, kind of recent de novo design papers that came out, one with mini proteins, one with antibodies, and they're really targeting exactly the same epitope that Ariel was targeting 15 years ago now. And it's a tremendous accomplishment that this can be done with way less work and, even that it can be done in antibodies now. But if you zoom in on the structures, this is the epitope Ariel was targeting. Tim Whitehead is here in the audience. He worked a lot on this project as well. And this is the epitope that, RF antibody is targeting, and even some of the interactions to the influenza are almost kind of exactly the same. So I think, there's been really tremendous progress in de novo design, but I think there's still a lot of unsolved problems in terms of how to bind to polar epitopes or, loopy epitopes or epitopes of different shapes. In contrast, natural antibodies are able to target these complex polar epitopes. So this is some data from Jim Wells's lab. And they looked at, basically they did a study of what do antibody epitopes look like. And so the first thing is that I'm showing here, right on the x-axis, you have all the 20 amino acids. And on the y-axis, you have how often they show up in antibody epitopes. And you can see that even charged residues like aspartate, glutamate, and lysine show up almost at least as much as in the natural antibody abundance and potentially even more often in antibody epitopes. So antibodies really are, targeting these polar epitopes. And more than that, this is still data from Jim's lab from the same paper, they're targeting, 50% of the time residues in loop regions. So there's really kind of I think an ability of antibodies to target these complex polar epitopes that I think we should, still think about how can we take advantage of this in the field. And this is sort of a simplistic view, but I think there's a few reasons that might explain why antibodies are able to do this. It's definitely not a complete explanation for why antibodies can bind to polar epitopes, but I think one reason that's come out in recent years for why antibodies can bind to these charged residues was provided by Steve Elledge's lab in 2023 when they put out this science paper showing that antibody germlines have some conserved motifs that allow them to bind repeatedly to charged residues, including things like arginine and especially lysine that are, really difficult to design binders to. And so I think this is like a really amazing thing that even though for us it's very difficult to design binders to these residues, nature has sort of created this solution that allows it to repeatedly get binders against something like lysine. And on top of that, of course, antibodies bind through these six complex loops, and especially, right, CDRH3, which is extremely diverse and extremely complex. And I think this sort of accounts for at least some of the reason that antibodies are able to bind to these structurally complex epitopes. And that's great. But the flip side of this is because antibodies are binding through these complex loops and they're doing these complex interactions, there's a lot more room, I think, to mess things up, right? And so this is why we need in vivo all this B cell machinery to make sure that we're not just ending up with misfolded or polyspecific or antibodies that bind to self proteins. And so one way to think about kind of the goal of this project is, from a very practical point of view, like we want to produce drug-like antibodies that are able to target these complex polar epitopes. Another way to think about it is we want to use all the design methodology that we've been developing over these years to try to control these very complicated loops that antibodies use to both get high structural diversity in these synthetic repertoires while also maintaining high developability and stability at the same time. Yeah, and so during the talk today, I'm going to talk a bit about the design procedure that we came up with to try to solve this problem. And I'm also going to show some experimental results from a proof of concept repertoire that we've built during my PhD. But I want to motivate the talk by saying that I think one of the really exciting aspects of what we've done is that these repertoires are really, we would say, algorithmically designed, meaning that we really know everything that goes into these repertoires, and we've selected what's in there very carefully. And so what we think this allows us to do is to start doing kind of really massive scale design test learn cycles where we could design, for example, a universal synthetic repertoire. We can build it and screen it for a function that we're interested in. So in my case, we thought about developability and binding to a few antigens, but you could imagine doing this for all kinds of different purposes. And then we can learn from these screens that we've done and go back and learn rules, governing certain functions, and then try to design sort of second generation libraries that fit these criteria or fit these rules that we've learned. And so I think this is sort of like, the dream that we're trying to build to with this project. And so I just want you to keep that in mind throughout the talk, that it's not only about these experiments that I'm showing, but it's also kind of about what we think this whole process can allow us to achieve. And yeah, so hopefully that sounds good. But the scale of this project is really quite huge. And I would say it's like close to unprecedented in size in the protein design community. When you're talking about designing large proteins, I'll give you two kind of benchmarks to compare against. These are both from our lab. There was a project where there were 1 million designed enzyme variants that came out 3 years ago now. Another project where there were 10 million designed GFP variants. And I think, again, this kind of set sort of benchmarks for how big you can go if you want to really design these large proteins, to have repertoires of large designed proteins. And we probably need to go quite a bit bigger than this in our antibody synthetic repertoires, synthetic antibody repertoires. But on top of that, the success rates in these projects were overall, although very high relative to, I think, some previous approaches, still kind of quite low. Like, so they're sort of typically below 1% successful designs, maybe getting up to kind of low single-digit successful designs. And of course, if we want these repertoires to really be useful, we need to go way, way higher than that in terms of how many antibodies are stable and functional. And to just give, kind of a generic sense of how high we probably need to go, this is a review that's already a bit out of date, but I don't think the numbers have changed too much. The column to pay attention here is on the right, where you can see the size of other synthetic phage display libraries, which range from something like 10 to the 10 to 10 to the 11. And so I think if we want to even do a proof of concept, we probably have to, think about getting up to the 10 to the 8th, 10 to the 9 scale. And to be really competitive with these repertoires, we probably have to think about getting to 10 to the 10. So this is really kind of quite a scale up over what was done before for full-length proteins. So to deal with these complexities, we split this problem into two kind of modular parts, following sort of, very typical breakdown that people use in antibody variable fragments. So this is a cartoon of IgG type antibody, right? It has this classic kind of Y shape. And at the top, you have the variable fragment, which is the part of the antibody that typically binds to the antigen. And you can, sort of conceptually break the variable fragment down into two regions. At the top, you have the CDRs, or complementarity determining regions, that typically do most of the work of binding to the antigen, with, of course, CDR H3 sitting here, typically, contributing a lot of the binding to the antigen. And these CDRs are really amazing because they allow you to bind to such diverse antigens, but because of that, they're quite diverse in both their structure and their sequence, making them, and of course, there are these solvent-exposed loops. They may be sort of flexible in some antibodies. So this makes them quite difficult to work with from a design point of view. At the bottom, you have the framework, and the framework actually still has quite some diversity in it, but it's certainly less diverse than the CDRs. It's probably easier to work with from a design perspective because there's a lot more secondary structure in the framework, and there's a lot less sequence diversity. But the primary challenge when designing the framework is the kind of the wisdom in the antibody engineering field would tell you that you can't make too many mutations in the framework away from the human germlines before you start having concerns about immunogenicity. And so during my PhD, we developed two methods to kind of deal with each of these regions, one called QMAB and one called LaFab. You can see the acronyms over here. And for the rest of the talk, I'll tell you a bit about each of these methods and show kind of some experimental results we have showing that these methods do something useful on smaller problems, and then go back to the library design. So I'll talk first about QMAB. Again, QMAB is really all about how do we design, in a sense, framework regions without going far away from the human antibody germlines. So the way we came up with to do this is we start by making a database of what we call acceptor frameworks. And the way we do that is we combine all the human antibody germline genes. So this is 2 genes in the heavy chain and 2 genes in the light chain. The D genes in the heavy chain we ignore for the moment. And this gives us 20,000 acceptor frameworks. And then if we want to optimize an existing antibody, what we do is we take the CDRs from the parental antibody and graft them onto each of these acceptor frameworks, which gives us 20,000 reframeworked designs. And if you think about it just sort of conceptually for a moment, the idea here is that we're maintaining the CDRs in all of these 20,000 designs. So we should ideally maintain the binding function to the antigen of the parental antibody. But we have all these different frameworks, and so we're able to kind
I'll
of
get
sort
him.
of
Yeah.
diversify this framework region. But since all these frameworks are coming directly from combinations of human germline genes, none of these antibodies are really much less human than any other ones, except for maybe in the junctions between the CDRs and the frameworks. And so that's sort of like the concept. But the thing is, what we know from a lot of work, especially on antibody humanization, is that this concept doesn't exactly work because the CDRs and the frameworks are not really modular. And there are interactions between the CDRs and the frameworks that are really important for holding the CDRs in place so they can properly bind to the antigen and they're not just kind of flopping around. And so most of these 20,000 designs probably would have low affinity and would probably also have problems with things like expression and stability. And so to get around this, what we do is we model each of these 20,000 designs on a structure of the parental antibody. This could be either an experimentally determined structure or a predicted model structure. And we rank the designs by their Rosetta energy, basically directly asking the question of which among these 20,000 frameworks can maintain or potentially even improve these interactions with the CDRs to kind of lock them in place properly to bind to the antigen. So we published this method in 2024. At the time, that paper was mostly focused on antibody humanization, but given some recent results, we've sort of been thinking about this method slightly differently than how we presented it in that paper. And so what I want to talk actually about today is unpublished results. This is a collaboration with Pete Tessier's group at the University of Michigan. If anyone is at PEGS this week, Pete is also at PEGS. And I think this is a really fun project for several reasons. So one reason is that we're working with kind of a fun antibody format. It's a 2x1 bispecific antibody. So the two Fab arms are identical, and one of the FC chains has a single chain variable fragment hanging off of it, which is the part of the antibody we were designing. And they use these shuttles for brain delivery. So this scFv arm binds to the CD98 heavy chain, which sort of takes the antibody across the blood-brain barrier. In this case, the scFv arm came from rat, and they wanted to do experiments in mice, and they were having problems with immunogenicity. So we actually wanted to murinize the antibody in this case, which was kind of a fun project for us because we had to rewrite QMAB a bit to work with mouse germlines instead of human germlines. But I think it also was nice because it highlights sort of, I think, part of the power of working with physics-based methods where it may be harder to do this with AI-based humanization methods. The design started from a bodybuilder 2 model structure, so it was nice to see that QMAB can work based on model structures. We've shown this already a few times with the paper, but it's always nice to see it again. And maybe the most interesting thing is the antibody exhibited high polyreactivity. So our goal in this project was actually not only to optimize the mouseness of this antibody, but also to lower the polyreactivity while maintaining all these other characteristics that we really care about. So we sent Pete's group 20 designs. In all the graphs you'll see in this section, the parental antibody is shown in gray all the way on the left, and the 20 designs are shown in these blue bars on the right. You'd always have this black dashed line that kind of just goes across and gives you a sense of the values for the parental antibody. And so here, this is ELISA data of the antibody for binding of the scFv arm. So most of the antibodies pretty well maintained the affinity of the antibody up to maybe 2 or 3-fold worse. Many of them were sort of really even with the parental antibody, and a few of them were not so good, but that often happens in design campaigns. And I just want you to focus specifically on these two guys here, designs 8 and 10, because they'll show up a few times in these different experiments. Pete's group then measured the polyreactivity of the antibodies. Here you have the value of the parental antibody, which is quite high. And here in a red dashed line, you have the threshold that Pete's group uses to classify antibodies as sort of good or bad. So you want to be below this red dashed line. You see that the parental is way above this red dashed line, and many of our designs were either below the red dashed line or even all the way down at 0. So that was, really nice for us to see, even though we didn't, sorry.
Maybe you'll get to that. What is your design objective in trying to lower polyreactivity? What's the optimization?
So that's kind of the interesting thing here is we didn't do anything directly to lower polyreactivity. Our hypothesis here is that I think polyreactivity is sort of an amorphous characteristic. It's a bit hard to design for, but what we think we can do is create different versions of antibodies that are quite different from each other while maintaining the same or similar affinity. So these antibodies, these designs we're showing, because they're coming from very different frameworks, they might have something like 40 or 50 mutations between one another, which results in very different patterns of charge and hydrophobicity on the surface. So the idea here is kind of like, we don't exactly know how to optimize directly for this, but we think we can create diversity and then screen in kind of a low throughput manner. Okay, so moving on, they then looked at a subset of designs and measured their thermal stability. And again, like kind of out of the 8 that they tested, 6 looked pretty good. 2 of them were a bit struggling. Oh, yeah, sorry.
So these the PSP scores that based on this 370,000 scFv data.
This is coming out of no, so this is coming out of a bit earlier work from Pete's lab, and so this is basically scaled from 0 to 1, where 1 is, I don't remember now exactly which control antibody, but 1 is an antibody kind of known to be highly polyreactive, and 0 is known to as an antibody that's known to be very good. So they always run it kind of in the same plate, and then they normalize the scores between the two. Okay, so most of the designs, including designs 8 and 10, had sort of good thermal stability. Yeah, sorry.
Just quick, if you look at this, Rosetta energy to find out which are like the lowest one, and then that value that.
Yeah, so it's not, so there's kind of two things going on. So we're looking for low energy, and we're also looking for diversity. So we look among the low energy designs, and then we cluster based on the sequences. Anyone else?
Yeah.
Yeah.
After measuring autoreactivity, were you able to correlate the computational matrix? It seems like some specific metrics are correlated.
Yeah, let's see. I think I actually have a hidden slide about this. Check. Yep. So yeah, so like Rosetta energy didn't correlate with this very well. And I would not say this is a correlation, but we went and looked at the PI of the variable fragments. And basically what you see is that when you go to a bit lower PI, all the designs are below the threshold. Higher PIs are a bit more iffy with some designs being below this threshold and some not. So we've also like been thinking about how to incorporate even something like quite simple like PI into design methods that we've been doing, but we didn't do that. This is retrospective, right? Like it's not something that we looked at before. Okay, yeah, and so yeah, just because designs in 10, I wanted you to kind of focus on them as I showed this data because they sort of did quite well across all of these assays, which is really nice to see. And going back to the question that was asked earlier, these two designs actually differ from one another by 48 mutations and have over 30 mutations from the parental antibody, just kind of showing, I think, the power of this approach that we can get so many mutations into these antibodies that really drive big changes in things like surface electrostatics and hydrophobicity without actually touching the CDRs at all, sort of potentially reducing this to what I think is an easier problem than trying to design the CDRs. Okay, so moving on to LaFab. Again, LaFab is sort of all about trying to design antibody CDRs. And so the way we thought about doing this, everything going on in this method is based on antibody-antigen co-crystal structures, which is definitely, a limitation. It's not always the case that you have an antibody-antigen co-crystal structure. But for us doing structure-based design, I think it kind of gives us a window or like a handle to hold on to think about, the best way to move forward with these things. So the first thing that we do is we try to identify positions in the antibody that are making interactions crucial for stability and foldability. And the way we do this is we look at all the antibody CDRs and mutate them to alanine and then score them in Rosetta. And anything that gives a big increase in Rosetta energy, we hold fixed. But we know that Rosetta oftentimes sort of undervalues polar interactions that might be really important for the foldability of these antibodies or sort of the ability of these antibodies to fold uniquely into the shape that they're supposed to fold into. And so for that, we also do an alanine scan where we only look for increases in the hydrogen bond energy terms in Rosetta. And any positions that fail either of these filters based on these scans are immediately held constant. From there, we have a design process that combines Rosetta atomistic calculations and phylogenetic information that we use to design multi-point CDR mutants. And this follows quite closely to the FuncLab approach that was published from our lab in 2018, which is mostly used to design enzyme active sites. And yeah, if you want more details, this paper is now on bioRxiv. So just as a case study, so I'll show probably two case studies from this work. In the first one, we started with an antibody, which is an anti-lysozyme antibody, which is obviously not the world's most interesting target, but it is a really nice model system to work with. And it allowed us to compare, to past works and things like that. we designed a library of 7,000 multi-point mutants. And again, every variant in this library was explicitly designed, which was screened using phage display by Adva Michali, who is a scientist at the Israel Institute for Biological Research and was doing a sabbatical with us at the time. After a few rounds of phage display, we selected 4 colonies for biophysical characterization. We really didn't do anything like very smart here. We just sequenced 20 colonies or so and just tried to pick a few out of those 20. So we expressed those 4 designs as IgG1, or actually IgG2As because it's a mouse antibody. And first we checked their, the affinity according to SPR. Here you have the parental antibody in gray on the left. And so all of the designs had improvements in affinity, with the best design having about a 30-fold improvement in affinity down to around 50 picomolar, which is really nice to see. But, of course, it's not particularly hard to affinity mature antibodies using yeast display. But what we were really excited to see is that the 4 designs also improved expression in CHO cells and improved thermal stability of the antibodies. So we were really kind of in one design screening round able to optimize this antibody for several characteristics. In addition to that, these 4 designs had 6 or 7 CDR mutations from the parental antibody and 3 to 5 mutations from one another. So it's not just the same solution over and over again. And we're able to, I think, design quite a big number of mutations into CDRs at one time. Seeing this sort of success, we wondered like if we could get a better readout on, how accurate is this design process, not just looking at 4 designs, but looking at the whole library. And for that, we turned to deep sequencing. These are logo plots of the unsorted library and the library after 3 selection rounds. And so there are 9 design positions in the library shown here. These are the parental identities at the bottom. And there's a few things I want you to notice from these plots. One is that even after 3 rounds of selection, the library didn't really converge much. There's still quite a lot of diversity in the library, which we were excited to see because we think that, suggests that the design was pretty accurate. There are a few cases where we had mutations that were quite successful. So for example, here, there was a valine in the parental antibody, which is pretty much gone after selection, suggesting that, our design mutations were much better. And the same story here with an asparagine in the parental antibody that's pretty much gone after selection. On the other hand, there were 3 cases where we had mutations that didn't do so well. And these were all mutations actually from serine to something else. And some of these are, not really drastic mutations. I mean, it's a serine to threonine. The threonine was really counter-selected here. Here we had a serine to asparagine, and here a serine to isoleucine. So we went back to our models and tried to understand like what went wrong here, right? Because all of these scored really well according to Rosetta energy. And we saw basically 2 trends. So one thing is this is a serine to asparagine mutation. This is on CDRH2 right next to the antigen. I don't know if you can really see it well here, but when we made the mutation in Rosetta is actually changing the backbone a bit to accommodate this mutation. And obviously, I don't think this fractions of an angstrom change in the backbone is the problem, but I think this is reflective of some kind of structural instability in the structure that Rosetta is trying to resolve by doing this and is somehow able to get to a good score. But yeah, it's probably representing some kind of misfolding that happens in the antibody. And then there was a similar story here with a serine to isoleucine mutation. And so this is something that really surprised us actually, and we've now sort of started incorporating these things into a lot of design that we're doing, like really being sort of wary of Rosetta changing the backbone to accommodate mutations. Although sometimes, of course, it's necessary. So it's kind of a double-edged sword. The other deleterious mutation we saw, and I just wanted to show this because I think it's like really amazing, is it's a serine to threonine mutation on CDRL3. And this serine is making a hydrogen bond with a histidine across the framework. And even though you sort of think like, yeah, it's just a serine to threonine mutation and it should still make this hydrogen bond, it's really, completely destructive. And so I think that just really highlights like how hard this problem is and how much detail we really need to look into to really understand what's going here. And to be honest, this was a position that was flagged by our automatic screening, and I added it back in my infinite wisdom. But it was, I think, actually kind of a stroke of luck because it really showed us, how sensitive of all this is. The second case study I'll just go through quickly. This is a collaboration with Mark Craig's lab at the University of Southampton. And basically here we wondered if we could get similar effects not going through library screening, just testing a handful of designs. And here we started with an antibody, which is not really very good affinity. This is SPR with the IgG flown over immobilized antigen. So this 26 nanomolar is really not very good affinity. Nevertheless, through testing only 10 designs, which all had 6 mutations from the parental antibody, we were able to get designs which were quite a bit better than the parental antibody. Again, something like a 30-fold improvement. We did then a whole characterization of the developability of the design. So we first looked at expression. The designs might be a bit better than the parental antibody on the left. I'm not sure. It's only one data point. It's hard to say. But when we look at thermal stability, the designs look really good compared to the parental antibody. And the same if we look at things like self-association. So here, this is size exclusion chromatography and AC SINs. This is, a bad control. This is a good control. So the parental and the two designs look quite fine. And this is for non-specific binding. This is the PIA ovalbumin kit. So it's a depletion assay. So a higher bar is better. I know that's a bit confusing. But again, the parental and the designs all look good. So again, we were really happy to see that it looked like we were able to optimize affinity quite a bit without degrading other characteristics. Yeah.
Said before, you were mainly going for diversity for selecting the designs to test. Here you're only going down to 10. So I'm wondering if you're doing any additional filtering or any other metric ranking to account.
Yeah. So it's again, so there are two, it's like two different methods, and you always kind of have different things that you're looking for. Here, because we're working with CDRs, you're not, you're much more limited in the numbers of mutations that you can make. So we're only getting 6 mutations. When we work with the framework, we're getting 50 mutations potentially. But we are kind of making sure that all the designs have at least a few mutations from each other. So basically what we do is, we rank all the designs by Rosetta energy, and then we take the best design and then say, okay, give me the next best design that's at least 2 or 3 mutations away. And then give me the next best design that's at least 2 or 3 mutations away from both of those two. And then you just go down the list. Yeah, except for, when we did the library screening, then you're just like throwing everything you can into the library. Yeah. Thanks. Yeah.
So, like you burning with parental antibody, is there an interest structure for this at all?
In both of these two cases with the CDR designs, there were. In the framework design case, it was a predicted structure. Of course, needing a antibody antigen structure as a starting point is a limitation. Like there's no doubt about that. We've been thinking about, if there are ways around this. But the thing I would say is that, keeping in mind that a lot of the motivation for this project was really to practice designing antibody CDRs for the bigger library stuff that I'm going to get to. Yeah.
You talked about the beginning.
Yeah, we try to get rid of them. As best as we can. Like sometimes you see in the parental antibody that there's a PTM liability that looks very difficult to get rid of. But we try not to add new ones in the design process at least. Okay. So yeah, after this sort of like, long-winded road, I want to get back to kind of the main plot line here, which is how to design so many developable and structurally diverse antibodies. And so the way we did this is we started with hundreds of structures from the protein data bank. Onto each of these structures, we modeled thousands of germline gene combinations, keeping the H3 loop the same as in the parental structure. And we then ranked all of these models by Rosetta energy, giving us, whatever, hundreds of thousands of models that we're ranking. And so I hope you can appreciate that this, first of all, looks very similar to what we did in QMAB, and the fact that we had some sort of confidence that the energy ranking that we're getting from QMAB, which is an automated method, was doing something useful, enabled us to sort of go to this much, much bigger scale and do this like straight away on hundreds of structures. And of course, it's not perfect, but we have some confidence that these energy-based rankings are giving us something useful. But now we don't want to select just a few designs to experimentally validate. What we actually want to select for are what we call sets of designs, where we have different sets of germlines and structures where we can recombine within one set all the germlines on all the structures and have a high percent of models that come out of this with low energy. And the reason we want to do that is because then within each of these sets, we can assemble all the antibodies together with one pot cloning, which just makes our life way, way easier down the road. And so to do that, we used a modified version of a method called EpiNet, which came out of the lab and was used to design these GFP and enzyme library papers that I talked about. Yeah, which I'm happy to talk about later, how that works if people want. I think one of the really cool things about this strategy is it leads to really large changes in the surface electrostatics and the shape and the kind of binding pocket of the antibodies that we get out without going at all essentially away from the human germlines. So what I'm showing you here is these are model structures. This is one structure that we used in the library. You can see the CDRH3 is sort of somewhere like here. So you're kind of looking down into the antibody as if you're the antigen. And this is two V genes modeled onto this structure, a light V gene and a heavy V gene. And if we then look at the same two V genes modeled onto a different structure, you see that the structure is different, and especially the H3 is in a different conformation, but the pattern of electrostatics looks kind of more or less similar. And that makes sense because the sequences are really close to the same, except for CRH3. But if we then play the same trick on with two different germlines, again, this is all inside one set that we've built, right? And all of these are low energy structures. We see that going down the columns, the structures are kind of maintained, but this pattern of electrostatics is completely different. So these are probably like very different functional antibodies. Again, even though we can recombine these all versus all and get something like 90% antibodies that we're labeling as low energy and foldable. And the other thing I think is cool about this is the designs are fully germline outside of CDRH3, meaning that one, we get to benefit from, some kind of things that are known about germline antibodies, that they may be kind of pretty stable. And on top of that, we're maintaining these grab motifs that we know are present in germline antibodies, meaning that we may get binding to these sort of polar or charged residues that are typically very hard to get binding to. Having selected these sets, we then did a massive design project on the CDRH3 loops of all the structures we selected using LaFab. And so what you see here is every time there's a residue in red, it was held fixed by our LaFab automatic calculations for hydrophobic interactions. In pink, it's held fixed for polar interactions like these hydrogen bonds here. Everything in gray, this is a surface representation of the rest of the variable fragment in the back, and everything in gray, it was sort of available to be designed, with the blue being mutations that we actually designed, including some pretty drastic mutations like a glycine to arginine mutation. And then if we look at a different H3 in the same library, we see that the pattern of mutations that we have to hold fixed is like completely different, and the pattern of mutations that we're getting are completely different. So this is why it was so important to us to have this automated method that, again, we'd sort of validated on other problems and had a sense was kind of doing something useful. So now we had all the sequences that we wanted to design, and we had to figure out how to assemble this giant thing. Sveta Gaiduk, who was a technician in the lab, helped. Yeah.
So I couldn't help but notice the previous slide. Some of these are pretty rich in aromatic residues. Do you have any sort of filtering to try to make them not too?
Yeah. So at the time, we didn't. Now I think that was a mistake. And yeah, like going back, yeah, I think in future rounds of this, that's definitely something we'll look into a bit more. But also we sort of wanted to do this kind of like, a bit, I don't know, loose isn't the right word, but kind of like see how things go and try to, because again, we're really trying to not only build one repertoire, but kind of like learn things about how to build better repertoires over time. So the way we decided to assemble this thing in the end is using this one pot Golden Gate cloning. We split this right into 5 parts, the 4 germline genes, which we ordered as cloned genes from Twist, and the H3s, which we ordered as oligo pools as Twist. Could then assemble all of this using Golden Gate cloning, which sounds really easy, but took quite a lot of time to figure out how to really get this to work reliably. And once we have that, we can transform it into bacteria and then go straight, to these phage display, or if somebody wanted, they could move it to yeast display pipelines that, we all know and love. We assembled this repertoire from the time that we got the DNA, once we've kind of figured out how to do everything, in about 1 week for less than $30,000. $30,000, a lot of money, but for 500 million designs, not too bad. We screened it against 4 targets. We found binders against all 4 of the targets and multiple binders against 3 of those targets, despite testing less than 50 colonies for each target. Among the 13 binders we found, we expressed 9 of them as IgG1s. This was expressed by Twist. This black dashed line is showing you the average expression level of IgG1 antibodies in Twist system. So you can see that most of the designs are way above the average. 2 of them are sort of average, which I think is not too bad for the worst 2 designs. We then checked the binding of the designs using ELISA. These ELISAs were done by Rebecca Weiland, a postdoc in the lab, together with Sveta. As you can see, these are EC50s compared to 2 control antibodies. Most of the antibodies we're getting out are sort of like single-digit nanomolar EC50s. Maybe some are below. But of course, lysozyme and RBD, again, they're not like the world's most interesting targets. They are nice because people have worked with them before. Having seen those results, we moved on to 2 kind of more interesting targets: mouse FLT3, which is a cytokine receptor, and a solubilized version of the Lujo virus spike protein. And again, we're getting sort of like low nanomolar EC50s, which are, not super high affinity, but kind of reasonable, I think, to get out from 1 round of phage display. Rebecca then did what I think is a really nice experiment, where she took all the antibodies and tested them in a 3-point ELISA titration against all the antigens. What you see here is the area under the curve from this titration, showing that all the antibodies really bind specifically to the antigen that they're supposed to bind to. Which is really nice to see that, again, we didn't exactly optimize any of these antibodies for specificity from a design point of view, but we did, and we didn't do any fancy screening or filtering after the fact, but we did manage to get antibodies which were pretty specific. I kind of promised before that we're going to get out antibodies that are diverse in both structure and sequence, and looking again at these surface electrostatic maps, the same as we saw before, the antibodies really do seem to be diverse in both structure and sequence. Out of those 9 antibodies, we have 6 different PDB structures that the antibodies are based on, and really different patterns of electrostatics, which was nice to see. We'd also said we were going to get developable antibodies, and so we again partnered with Twist to measure the developability of the antibodies. In all these experiments, the designs are in blue on the right, and they're compared to a panel of 12 therapeutic antibodies in gray on the left. So this is first thermal stability, so the designs were pretty good in terms of thermal stability. This is based on nano DSF. The worst one is about 69 degrees Celsius, still quite good. And they're also quite good. This is a melting temperature. They're also quite good in the temperature of aggregation onset. And this is really nice for us to see because, again, this stability is really the thing that we were optimizing for here by the design protocol. Twist then did a whole panel of developability assays using things like cross-interaction chromatography, hydrophobic interaction chromatography, BVP ELISA, which is supposed to test for nonspecific binding, AC-SINs, and DLS. And the way this works is that in each assay, the antibodies get scored. 0 is like they pass the assay, they look good. 1 is an intermediate flag, and 2 is a serious flag that should really be worried about. And then these scores are summed across all of the assays. So this basically is something that goes from 0 to 10. Nothing got a 10. So compared to this panel of therapeutic antibodies, most of the designs, I think, did pretty well. There were 3 designs that definitely had problems, mostly with things like hydrophobicity, kind of going back to Chris's question. So we then went back and looked at the design models, and all 3 of these antibodies had exposed either tryptophan or phenylalanine on H3. You might ask, Chris did ask, like, why did these end up in the design to begin with? And honestly, I don't really have a good answer for that, other than to say that, we're going to avoid it in the future. And I think this is a really nice example of how, because we're designing these repertoires, we can then just go back and say, like, okay, if we want a more developable repertoire, we just avoid this in the future. And that's sort of like a small scale example of how we think this can work, but I think this could work also on a much larger scale. It talks about this grab motif thing, and we're really interested to see if any of the designs bind through grab motifs. We're also interested to see if the structures match to what we expected. So together with Roman Kramishinsky, who is a staff scientist at Weizmann that works with cryo-EM, and in Bar, a master student in the lab, also helped with this. We solved the cryo-EM structure of one of our anti-lysozyme antibodies. This antibody has 57 mutations from the structural template from the PDB, so there's a lot of mutations there. And you can see that the structures overall agree quite well. The starting antibody here was not a lysozyme binder, just to be clear. There was a bit of like swiveling in CDR H2 and H3 relative to the structural template, which I think is actually nice to see that we're not overly rigidifying the CDRs, right? Because we do want these repertoires to be able to bind to diverse antigens. What we were really happy to see, yeah, it might be a bit hard to see this here, I apologize. So first of all, this antibody binds to an aspartate on the lysozyme through a grab motif that was identified in the Steve Elledge paper from 2023. I think really highlighting, again, the power of working with germline, antibodies that are quite similar to germline. But on top of that, the antibody actually also binds to a second lysine from the antigen. And this is through what we think is a new grab motif that was not identified in the Steve Elledge paper. I think sort of showing that this is like, what I think is really cool about this is it's like a library now that I think we can start, in addition to the practical like uses, start asking all kinds of like interesting basic science questions about, what's going on in antibody development. And yeah, so we think it's really cool that we got, a designed antibody that is binding through these charged residues. And of course, this results in a pretty high electrostatic complementarity between the antibody and the antigen. So in conclusion, I think that, structure and energy-based design is an excellent first approach for designing and optimizing antibodies. By no means perfect, but maybe a really good place to start. Even at the scale of billions of antibodies, I'm really excited, in the future about thinking about how can we combine structure and AI-based design to enable improved design protocols. I think the solution to this problem, is definitely somewhere in between. We're really excited about, this idea of how can we use these repertoires to learn things and then generate improved repertoires in the future, or repertoires tailored for specific functions, which I think is, a really cool application. I think we've shown in like really small scale that this might be useful, and I'm really excited to see how the lab continues this. And lastly, I think these germline encoded motifs really provide a path for targeting polar and charged epitopes that's very difficult, very difficult to design binders for. Yeah, and I'm like personally really excited and thinking about like how can we use these motifs in all kinds of different ways. With that, there's lots of people to thank. I think I got all the big ones. Of course, everyone in the Fleischmann lab, and especially Ariel for all his mentoring over the years. This is like one of my last opportunities to put on my Fleischmann lab hat and give a talk as a member of the Fleischmann lab. So I was really excited for that, and thank you for the time, and I'm happy to answer any more questions. Yeah.
So, for libraries that are displayed on surfaces, for example, like antibody libraries, it's kind of, I don't know how well known it is, but it might be very well known that only a small fraction of these display sequences actually do display, or they don't, many don't express.
Yeah.
Is the point. And my question is, do you have a feel for how many of your 500 million sequences actually do display on the surface of phage?
Yeah, so it's a really, really good and important question. And so I can tell you on a very small scale. So, we've sequenced colonies after the bacterial transformation and then after having gone through the phage. And not a huge amount of colonies, but we don't really see any evidence that like the diversity is crashing out. That doesn't exactly tell you that they display on the phage, but it tells you that they at least make it through the phage. one of the reasons we display this library as fab, and one of the reasons that we wanted to do that is you could then think about doing selections with things like protein G and sequencing then after selections with protein G, and those are ongoing. But I don't have the results yet, but they're definitely things that we're thinking about, and I think that would be super exciting to like although I hope that we would find out that almost everything displays well, I think it would be super exciting to find out, hey, this portion isn't displaying well. Like, let's go back and design things to do better in the future. And maybe that, even helps us learn something about antibody stability and function.
I have two questions, one more specific, one high level. The specific one is, could you comment on the clustering of sequence-based clustering of the framework regions? Because it's like if you have framework regions but only like 30 mutations, then you still probably have like a high sequence identity between the combinations.
Yeah, so actually it's very simple what we do. We get the germline genes from the IMGT database, and the IMGT database are already classified on several levels. So there's what they call subgroups, which you could kind of think of as families, and then there's genes. And so we only take, like, we model all the combinations of genes, but then we only take the best combination of heavy chain and light chain subgroup. So we only have like one combination of genes from the heavy chain and light chain subgroup. So and these groups are defined based on sequence similarity, but that's how we do the cluster.
And then the second is you touched on like potentially a new grab motif. Were there other like biological kind of discoveries that you found, or if not, like what are some things you're excited to look?
Yeah, I mean, so I think searching for new grab motifs is really exciting. I know, Steve Elledge had found something like 20 in his paper. I think there may be a lot more than that. And I think this sort of library is a really interesting way to look for those because you have the same germline genes with many, many different H3s. And I'm sort of like starting to think that we might be able to look at things like, can we sort of understand like preferred V gene pairings based on the modeling that we're doing and stuff like that. I don't know if that really answered your question.
Thanks.
We're saying these as pH, HJ, LJ combinations on PDB antibodies. Then you go to those that are in place and clashes basically. So I'm curious, did you look when you starting antibody from the PDB, which has its own pH combination? Do you actually then just end up finding that same combination? Or ask the question another way around this, should write into clashes with the LPH with the so how does it follow?
Yeah. So one thing that I glossed over, or actually didn't mention at all, is because different VLs and different VHs have different CDR lengths, and so modeling, we really don't want to get into kind of trying to design new backbones of CDR loops, and so we're only modeling germlines that have the same CDR lengths on each PDB. So this automatically kind of like restricts you to some extent in the diversity that you can do. And of course, if you have a germline gene that's very similar to the antibody, it's going to be easier. But we're getting a lot of diversity onto each of these antibodies, and of course, keeping in mind that all these antibodies from the PDB have mutations in the other CDRs, so you're never getting like exactly the same sequence. We didn't look actually at, I mean, it's a good question, like what's the kind of like average number of mutations that we're getting or something like that, but I would expect it would be at least above 20 or
Actually, combinations of mutations.
oh yeah, so that
Given by that the LPH. I'm going to new ones. So do you actually get it?
yeah, I think we do. Maybe not in every case, but I think in many cases we're really getting kind of new combinations coming in. Yeah, Tim?
Let me ask you. So you were able to go and predict things that did not perfectly flash, give it a status and duration freeze, right? You should be able to predict incompatibilities of frameworks with a given CDRH3. That's the case, then you should be able to predict the like structure of CDRH3 given an ensemble, say, developable grid, by cascading a set of 10 frameworks, some which are incompatible, some the CDRH3. Have you guys considered that experiment? Or is that?
You mean testing them? You mean testing them experimentally?
Yeah, so let's say alpha seeded kind of like these series, right? You can design an experiment where you have sequences incompatible with each of the other ones compatible. So you can get the likely sequence function from just that experiment. So,
Yeah, no, it's an interesting idea. We haven't thought about it. I mean, everything that, so our whole kind of like thrust in this project is that we think it's very hard to, kind of design new loops. And so we're really trying to utilize like as much of the diversity of the loops that we have already on the PDB. But yeah, it's an interesting idea.
yes, I mean, the core layer is complex stuff. Like file structure and break down by frameworks and out of the model. It's $20 a pop. It's very.
Yeah, that's an interesting idea. Yeah, Chris?
I think it's remarkable the hits that you got out of sequencing like 40 or 50 colonies only. So I can't help but wonder what else is there. I wonder if you thought about like how would you, for how much deeper did you go in the diversity? Like what sort of, are your screening strategies bottlenecking the diversity, or could you possibly even, is there like altering how you screen it, find more interesting, useful things?
Yeah, so I think, so, we've thought about, like, I think the easiest thing to do is to like, deep sequence throughout the selections and get away from this colony testing to begin with. And we've thought about it, and it's something that, yeah, we're working on. I think, like, if I'm being honest, that in the current like iteration of these repertoires, we're not getting so many binders to given targets. But we're working. I mean, so I think the next kind of thing to do is, and this is like 500 million. Again, it's like, probably 10, 100 times smaller than other synthetic antibody repertoires that people are using. And so the next question is like, can you scale this 10, 100 times bigger? And can you start using really different frameworks? So could you do like a Lambda library? Could you do other kind of more diverse frameworks, which might give you more binders? This is why, I just wanted to kind of like reiterate that it's like a proof of concept. And I think there's a lot of things that should be developed in the next, couple of years coming out of the lab.
Library wondering polyreactivity of it's at all.
Yeah, so we did, this was, let's see if I have it. Yeah, this was a sort of one of the developability things that Twist did for us. You can see it here in either CSI, wait, yeah, anyway, there's a couple developability assays here, which have to do with non-specific binding. And yeah, especially the antibodies that, had these exposed hydrophobics have a bit of issues with non-specific binding, but a lot of the other antibodies are doing pretty well in like baculovirus particle testing and things like that, which, are not perfect, but.
Let's thank Ariel.
Thank you.
Fantastic. So I think there may be 1 or 2 beers left. So please, well, either way, please feel free to hang out, ask Ariel more questions, and we'll see you back across the river for Foster Birnbaum on June 10th, I think. So it's second Wednesday. Yeah, from 80 flag. So yeah, it should be great.
It was a nice talk.
Thank you.